So this is the most important aspect. This is why we do all this. It's not to do animal studies, not to do pharmacology studies, not to just make stuff because chemists like making stuff. It's to get it into the clinic. And the beauty of having a company run by a clinician is that's their focus. So we're going to talk about the IND a little bit, and then we're going to go through the 3 phases, and then I'll have some final points to consider. So this is essentially the index for an IND. And aside from the kind of generic stuff, you really want to have in your IND a clinical investigation plan, an investigator's brochure, the clinical protocols. There's a big section for chemistry, manufacturing, and controls known as the CMC section, you might imagine. And early on, the FDA is very interested in this because this is the first time you're going into humans, and they want to see that you're not manufacturing stuff that they'd rather not see go into humans in terms of purity. There's a pharmacology section, and then most importantly, that toxicology section. They want to see metabolism, some PK assessment done in animals. Any previous human experience. So if the drug was used by someone else and you're repurposing it, they're going to want to know a lot about what was experienced before with humans. And then there's other additional information. I'm never quite sure what this is. I assume if the drug was a street drug and now you're repurposing it to a psychiatric drug, they probably want to know some additional stuff about what was known about the street drug aspects of it. And you submit this to the FDA. There's what ours look like. Now they're all electronic, but you get a feel for number of folders, and it's usually done in multiple copies. So we were sending books or boxes and boxes to the FDA.
I'm sorry, what's the IND? One file or the entire?
The entire thing.
That's it.
That's it. That's an IND.
Can't you just send them a hard disk?
Now it's all done electronically.
Oh, okay.
Remember, this is back in 2000. So once the FDA gets this, a clock starts, actually, the relatively recent law where we pay for stuff and they're required to move it along. They have 30 days to respond. And after 30 days, if they haven't responded, you can go. You're free to go. You never get a yes out of the FDA. You just get a no. What we got was well, you know, that vinyl sulfone, you haven't really made it and demonstrated that you have it. So why don't you make it? And we have some questions about stability, and there were a couple other questions we had. Oh, the questions about starting dose in the clinic. And so we made some adjustments. We sent a letter back to them saying, yes, we'll take care of this. But none of them was a showstopper. And you don't want showstoppers because chances are you've told investors you're going to be filing, you're going to be starting your clinical trial soon. And if the FDA says, wait a second here, this tox is inadequate, you can't start, that becomes an announceable event for a small company, and nobody likes that. So on day 31, my boss on day 30, my boss and the chief medical officer called me in the late evening, and they were screaming at me. And they said, our regulatory person, who was a consultant, wanted to write back to the FDA or call the FDA and ask them, are we okay to go? And didn't I think that was stupid? And I have to admit, you know, you don't want to ask the question, you don't want to ask the FDA a question you don't know the answer to. Because they could just as easily say, well, we thought about it. So, no, we didn't call them back. We just went. We started the trial. So again, here's one of those generic slides on the various trials. You've all heard about these. You heard about them yesterday. Now, what I'm going to tell you as I go through these things are, you're going to say, you know, all those questions that Rita mentioned yesterday, were they really important? Do I really need to think about them? Well, when I'm done here, I think you'll know the answers to that question. But anyhow, here are the 3 phases. We talked a lot about phase 1 yesterday. We'll talk a little bit about it tomorrow, a little bit on the next slide. These are the general cost considerations, and you're pretty much looking at tolerance and PK and dose schedule. Phase 2, much more expensive. It's all about seeing if you can get a hint of efficacy. Fortunately, in oncology, you go into patients. In fact, I have a hard time going into normals. I mean, I have an ethical issue with that. I don't think you should go into normals ever. I think you should always go into patients because you're ultimately going to treat patients. You want to know about the safety in patients. And by the way, there's a chance the patient will benefit. And being in oncology, that's kind of important because the patients that we see in phase 1 are all end-stage cancer patients. So you look for some biological activity signature in phase 2 and hope it guides the design of your phase 3 studies. And then your phase 3 studies are these extremely expensive studies that can last 3 to 4 years or longer. And really what you're doing is proving the drug is both safe enough for the intended population and actually has a clinical benefit that the FDA and you have agreed to is a clinical benefit that registers the drug. So before I forget, this little book up here, First in Human book, that's an excellent book. There's a lot of all the statistics you saw the other day from Professor Liu. They're all there, plus there's a nice analysis of all these different kinds of ways of doing a phase 1 in humans. Remarkably concise and good book to read if you're interested in such things. So we did a 3+3 dose escalation design, modified Fibonacci. 2 things you heard about yesterday. As a chemist, I understand 3+3. It's very simple for me. And I actually like the trial design because it was so simple. And I understood it. It's a little rubric that you just kind of follow. And at the end of the day, you end up with a dose that everybody's comfortable with. Now, I understand all this other stuff's come in and makes it more efficient. And I would like to actually have that discussion at some point. If I had done that kind of study with this drug, would it have been a good idea? Because it turns out that because we started at such a low dose, the FDA was right, it took us forever to recruit, to get to the dose-limiting toxicity. It turned out the dose-limiting toxicity pretty much aped what we saw in the dogs and rats. It was hematuria. It was reversible, so that was always nice to know. And then we saw a lot of grade 2 stuff that you tend to see with cytotoxics. Nausea, vomiting, fatigue, and some anemia. The dose turned out to be remarkably high. So people are indeed more like rats than dogs, at least for this drug. And it took forever to get there, and now I'm in charge of production. I'm supposed to be supplying this drug, and I'm supposed to be supplying it to patients. Imagine how much drug at this dose, up to 2 grams a patient, I'm going to have to provide to do a clinical study. And this was a very tough early stage process development and manufacturing project for a lot of reasons. When we looked at patients in our phase 1, we had a lot of different kinds of cancers, and we saw a lot of positive biological responses. Efficacy is an FDA word. These were positive responses, and including one case where they were on the drug for 9 months.